WIM8 can block these, but then the organizer expression factors can block BMP and WIM8.
Okay, then what happens?
Then the pharyngeal endoderm produces Cerebeus, Dickoff, Frisbee, Tiki, IGF.
Cerebrus, Fizzy, Dickoff, Tiki, IGF,
Yeah.
right?
And those block WINT. The head mesoderm and the quarter mesoderm produce noggin, cordon, and follistatin, which blocks BMP.
Oh, IGF and Cerebrus, they also block BMP.
Yes, sorry, I always
right,
forget
yeah.
those two. They all block BMP. With both WINT8 and BMP blocked, that forms the forebrain and midbrain. And with WINT present and no BMP, that becomes the spinal cord.
Okay, then,
And then when there's none of them blocked, that becomes the epidermis, and that is always blocked.
Okay, so that means we talk about head, tail, and we said like how all those things are formed. Now we are moving to the early embryonic development in mammals, okay? So we're going to talk about early mammalian development. Now, think about when you talk about fishes, amphibians, they're all laying egg in water. There's enough water, no chance of drying out. You talk about and water also actually protects because embryo cannot be damaged. You know, there's everywhere water there. You talk about the fishes, reptiles, they have kind of an egg in the shell, right? A lot of yolk there, a lot of food there. And again, the shell protected. And a lot of fluid. When we talk about mammalian egg, there is no such protection. For that, you have an amniotic sac, which is full of water, right? So, and then, but they don't have any yolk, no food. So it means they need to develop and they also need to get food. So, the biggest problem with the human developmental studies is that human egg, or the mammalian egg, I should say mammalian egg, is the smallest. Its size is only 100 micron. And Xenopus eggs is, actually, if you talk about Xenopus egg is 10 millimeter. So this is the size of Xenopus egg, and this is mammalian egg. So this is a huge difference of the size also. Now think about the number. We said 5 to 7, primordial follicles start growing out. So if you do artificial ovulation also, you cannot get more than 10, 12 eggs at one time. So that's also a difficulty in studying. Now the main hurdle is in studying the human or mammalian development is that embryo develops in other organism. That's the biggest hurdle in studying the mammalian development. And think about that, okay, you can do, that when we talk about in vitro fertilization, you can in vitro culture the egg, of course, you can get those and culture those, and you can keep them growing till the blastocyst stage. But after implantation, how to study? You cannot study humans after implantation. I will talk in a minute how it is difficult, but also you cannot tell a woman like 1 week pregnant or 5 weeks pregnant to give the embryo so we can chop in the section and study what's happening, right? You cannot do that. So those are all hurdles there. So what we know and how to circumvent those hurdles is stem cells. We'll talk about stem cells when we'll go there. But still, it's very difficult. So even in the mouse, it's very difficult to study those things because those are implanted there, right? So whatever we know is basically from the mouse. Early human development we know, but we don't know much about what is happening in the mammals. You may know from some aborted fetuses in the late stage, but really what is happening is mainly based on the other animal studies. Okay? So then, how this thing starts? We know that the first thing which is happening once the cell division takes place is the cleavage. That's the first thing. Now, another important thing is the cleavage, we said, starts immediately after fertilization, right? We're showing that. And we know that when the fertilization takes place in ampulla, right? So what is happening here? So think about so this is the uterus. So this is ampulla, right? So this fertilized egg, which has started its cleavage right from here, has to come here and reach the uterus for implantation. Okay? Now, fertilization is taking place in ampulla. When the egg is fertilized, you have this fertilized egg, 2N egg, and you have this zona pellucida, still intact there. And out of this, cumulus cells are there, we know that. So what is happening, that the cleavage starts here in the ampulla and keep on going, it's keep on developing until it reaches here in the uterus where the implantation is going to take place. Now, the cleavage in mammals is slowest in animal kingdom. The slowest in animal kingdom. So usually it can be 12 to 24 hours apart. It's not like a divide, like we saw in the amphibian movie, like click, and they are dividing. So it can divide and then stop. Now, the first cleavage is meridonial. So this egg is there. So the first cleavage takes place. Then this cell divides into two. And they are attached, but loosely attached. They are not like really compactly attached. They're like this. Okay, that's the first cleavage, meridonial. So this is the first meridonial cleavage. Now second cleavage is interesting. What happens in second cleavage, in one of the cell, this cell is there, it divides meridionally. Okay? The other cell which is here, which was here, this cell divides equatorially. So ultimately what you get something like this. Okay? Now, this is called, so you get this. So one meridional, other is equatorial. Meridional, and this is equatorial. Okay. Question?
I just thought that the cleavages were opposite last time we talked about them. Like the naming, like I thought meridional was this way and equatorial was this way.
No, equatorial is divided from the equator.
Okay. I'm just confusing myself.
Okay, so what we are getting here, did I tell you wrong?
Well,
I'm trying to look.
yeah, 'cause in the,
We had the third
We had in
cleavage
the-
was meridional.
North pole, south pole is equatorial,
okay, we
right?
had, this was equatorial, then the second cut was right angle equatorial to that, and then the third cut you said was, meridional.
Okay,
I think the difference
Yeah, that
is
was what I'm
that one
saying.
has poles.
oh, I'm writing it opposite, okay.
Yeah.
Here,
Okay. It goes like this, change it. Okay. So what we get there is, now we are getting 4 cells, but 4 cells are looking very different from each other, right? So this type of cleavage is called rotational. We are getting rotational cleavage. Now, another big difference in the mammal is usually you can get in what we learned earlier. We said 2 cells give to 4 cells, 4 cells give 8 cells, 8 gives 16, right? So there is what we call it exponential growth in the number of cells in other organisms, But in mammals, what happened, not all the cells, they divide equally at the same time. So occasionally, you can have, odd number of cells. You can have 5, you can have 7, you can have 15. So not all the cells, they are dividing like this. Okay, now what happens? These are the major differences. Now what happens, another thing, which is another critical thing is that during these divisions, when these divisions are taking place, what is happening that parental chromatin changes. Number 1, histones are exchanged with new histones. Histones are changed with new histones. Okay. Second thing, what is happening there is that gamete specific DNA methylation is removed. Gamete specific DNA methylation is removed. And then the third is the new methylation patterns, methylation for totipotency and pluripotency is established. So, all the methylation patterns are basically removed, histones are exchanged, and new methylation patterns are formed. Now what happened? By 16 cell states, what happened by 16 cell stage, what is happening, that each of the cell is essentially hypermethylated. So, when that means each of the cell is hypomethylated, that means these are essentially pluripotent cells. And that means that is the stage set for the new development. So what is happening? So we are saying that a cell is there. Now, these are not free. These are surrounded by zona pellucida. That's the important thing to remember. In a way, everything is happening in the egg. So, we are saying that when that fertilization takes place, immediately after fertilization, one cleavage takes place, and that cleavage is equatorial. Okay? And that divides cells in two. Second cleavage, one cell divides equatorially, the other cell divides meridionally, and you get eight cells right there. 2, 2, 4. How many? 4 cells. You get 4 cells, but those 4 cells are divided in different orientation. And this type of cleavage is called rotational cleavage. Okay? Then we are saying that in the mammalian development, all those cells do not divide, their number does not increase exponentially. Some cell might decide they don't want to divide. So you get often odd number of cells there. Then we are saying that during this division, what is happening, the histones are completely displaced, new histones are formed, and the gametic methylation patterns are removed, and new methylation patterns are formed. New methylation pattern means when the DNA is hypermethylated, the chances are, not the chances, always going to happen that all the transcription will be shut off. When you have very specific methylation, for example, in skin versus neuron, that means skin cells are only transcribing the genes which are for skin, neurons are only transcribing only which for skin, which for neuron. But here what is happening, slowly the methylation patterns are completely removed. And when they are removed, we are saying that become hypomethylated. And that's why they are pluripotent, because each cell is open to make any type of protein. That means the transcription of any of the gene can take place. So every cell is the capability to become everything in the body.
So would that be totipotent?
Yeah, initially totipotent. I use here term at the 16 cell stage, it's hypermethylated for the pluripotent.
Okay.
16 is pluripotent. 8 is totipotent.
Okay.
So pluripotent and totipotent are different. So this makes sense. Any question other than that confusion of equatorial, meridional?
I'm still confused on the division.
Uh-huh.
Because, so in our, when we did like these notes, we said meridionus was this way and equatorial was this way. And now we're saying that it's meridional is this way and equatorial is this way.
So they are also confusing me now.
You
So-
were right the second time.
So let's come out of that meridional equatorial thing. So
Which was wrong last.
North Pole, South Pole. Division like this is, it's actually meridional. You're not dividing the equator.
Okay.
It's meridional, right? And I was right, you guys confused me right in the beginning.
When we were talking about this, we said.
No, you look back at those.
Last time we went over it, we said it opposite.
You said the
Okay,
first two.
so this is right.
Yeah,
So we are not dividing equator, right?
no.
So it's meridional.
Okay.
And when you are dividing the equator,
Equator's
that is equatorial. So whatever is there, correct that. So it means I was also confused somewhere. So that confusion should be now resolved forever. Okay? So if you are dividing equator, creating new equator, that's equatorial. If you're not dividing equator, that's meridional. Okay, so fix those things, what was original was correct, okay. Now, anything else here? So far it is okay? Okay, now what happens? Okay. So then, we are saying the stage is set for differentiation at 16 cell stage, right? At that time, it's become pluripotent. But before that, something happens. So there is a process happen, what we call is compaction. Compaction. So one of the most actually crucial event that happens in mammalian cleavage is called compaction. So what happens? So you have this cell 1, 2, 3, 4, 5, 6, 7, 8. Now these 8 cells are there. Remember, these 8 cells are not just there. Outside is here is your zona pellucida. So this is your GP. These are 8 cells. 8 cell stages. Okay. Now all these at 8 cell stage embryo, the blastomeres are arranged loose. That kind of loose arrangement there. Okay. So what happens that following third division, when the 8 cells are created, right? So they actually change their behavior. So 2, 4, 6, 2, 4, and then 8. That's the third division. When they become 8, immediately after that, they change their behavior. So blastomeres change their behavior. They change their behavior. And what they do? They start expressing cell adhesion protein E-cadherin on their surface. So that caused them to start expressing so these 8 cells are there, and what they are doing, they start expressing so this is the E-cadherin. So E-cadherin they start expressing. And when they have this cell adhesion E-cadherin, then what happened? The blastomere huddled together. The blastomere huddled together. And what they do, they essentially form a structure like this. Like this, like your neurosphere. You cannot discriminate one cell from other cell. So this process then that leads to what you call is compaction. Now you cannot separate one cell from the other cell. So this is happening at 8 cell stage. So at 8 cell stage this is happening. Now, this 8-cell stage compacted. So this is 8-cell stage compacted embryo divide, and when it divide, that give you 16 celled embryo called morula. Now, if you look at morula very closely, what you will see in the morula, again they are compacted, but if you can somehow do the cross section and all those things, what you will see that these outer cells which are here, they are very large. So these are large outer cells. And the inner cells, these are the small inner cells. Okay. Now, what is going to happen? So I'll just go over this, then I'll talk about the next thing, what is going to happen. So what we are saying that 8-cell stage cells are loosely arranged. Then something happened, and these cells start secreting PKH, cell adhesion protein, that cause these cells to huddle together, and basically a process occurs which we call compaction. So these cells are compacted that you cannot basically separate one cell from the other cell. Then these cells, up to 8 cells, they are totipotent, right? They can become anything. But then these cells divide once more and become a 16-cell embryo called morula. At that time, some cells are large. Those large cells remain on the outer cell, and then some cells are small, they are small inner cell. Now what happens? These small inner cells, which are the large outer cell first, these large outer cells, they are going to form trophoectoderm. And when we say trophoectoderm, tropho is from trophic factor. Trophic factor means survival factor, and embryo, developing mammalian embryo, get all the survival factor from the mother, right? So it means that these cells will contribute later to the formation of the connection with the mother, right? And these small inner cells which are there, these small inner cells, they will be changed into inner cell mass. They will change into inner cell mass. What is going to happen? Ultimately, these cells are going to arrange in a way that you will have the large cell like this, and in this you will have these small cells huddled together like this. And these cells will be called inner cell mass cell, also called ICM. And outer cell, these cells will be your trophoectoderm. Okay. Now at this stage, this is the morula. At this stage, this embryo will be called blastocyst. Okay, now. So, this is clear, just talk to each other for whatever we have covered so far, so that we are all on the same page.
Amphibian eggs have a lot of lack of protection, and humans only have fluid and no yolk, so they have to get the nutrient to the mom. One smallest eggs, or the other smallest eggs at 100 micrometers. They're developing babies in other organisms, which is the biggest hurdle, because for studying microbiota, it only studied to the blastocyst stage, and everyone else we know is mice. Cleavage doesn't mean after fertilization, and the egg must move into the uterus, or child uterus. And cleavage intervals slows from 12 to 24 hours apart. The first cleavage in neodermal, and they are still loosely attached. So the cleavage is one cell is neodermal and the other is equatorial, which creates rotational cleavage. Mammals also don't undergo exponential cell growth because their cells can divide at different times, which can create odd numbers. And then during this division, parental chromatin changes because the histones exchange with new histones, gamete-specific DNA methylation is removed, and new methylation for total-potency and pluripotency is established. By the 16-cell stage, each cell is hypomethylated, which is pluripotent and differentiation. And before that, there was a process of compaction. So at the 8-cell stage, blastomeres are arranged loosely. And then following the third division, as soon as they become 8 cells, the blastomeres change behavior as they express each adherent on the surface and huddle together and come back into an indistinguishable form. And our tedipod, we can divide into a 16-cell embryo called a morula, which is pluripotent. And there are outer larger cells, which form the trophobectoderm, trophic factor, which makes it survivable, and forms a connection with the mother. And the inner cells are small and are changed into an inner cell mass to form the blastocyst.
Question?
Huh? Yeah, I read that too.
Question? Okay, any question here?
Is it still 16 cells and then the blastocyst?
Hmm?
Is it like the morula, and is the 16-cell stage? And then-
More or less 16-cell stage.
Right, and then when we go to the blastocyst?
There will be more cells.
More cells,
yeah,
okay.
yeah. But remember, all those things are happening inside the zona pellucida, so you can
Right.
draw zona pellucida throughout.
Alright,
So zona pellucida is still there. Now, so we are saying that we have this we have these cells here, and then we have these small cells here. Okay? So these cells, what we call trophoectoderm, also called actually trophoblast. So these cells, what they do, they are going to form non-embryonic portion of the embryo. So far it was embryo, right? They are dividing with say 8 cells, then we said 16 cells. So far everything was embryo. But now when they become trophoectoderm and trophoectoblast, they are going to form the embryonic, means that part will not be the part of the embryo. Okay? So what they form? They form chorion. And what is chorion? Chorion is the tissue going to be the embryonic or embryo derived part of the placenta. So essentially they are joining embryo with the placenta. The chorion is that part. Okay. Now what is the purpose? To get oxygen and nourishment from the mother. Remember, there is no yolk as it was in the amphibian cells. And then other thing, this is the one thing chorion is doing. The other thing is what chorion is doing. Chorion is secreting hormones. And why this is secreting hormone? To cause mother's uterus to keep the embryo. To keep the embryo. So how it is doing? So it's modifying the mother's immune system. So what is doing? So mother immune system can take this foreign body there, right? Can secrete all the antibodies against this. So there can be rejection, like the tissue rejection occur, right? So these hormones, what they are doing, they modify the mother's immune system. So therefore, no tissue rejection. And in this case, the tissue here is the embryo. So the mother immune system does not think that this is something coming from out. So what is happening? This is happening very early, right? So the first divisions, like from 8 to 16, that was the division number 4, right? From 8 to 16. When 16 cell occur, you get that arrangement. That hey, the outer cell or larger cell, they are going to give you tropho, ectoderm, trophoblast. They are going to make the chorion. So right here in the first division, what is happening? You have number 1, generated cells. Those are going to be chorion. That means no more the part of developing embryo. They are no more the part of the developing embryo, right? And then you have the second thing is formation of the cells. Those are going to develop in fetus. Two things have happened right there, right in the beginning, right? Now these cells which are outside, they are not going to contribute anything for the new fetus. Only these cells which were inside the smaller cell, they are going to give you the everything of the new fetus. Now, interesting thing is, this cell can give you everything, but not the trophectoderm. So this cell, the inner cell mass, this cell, ICM, cannot form trophoectoderm, but will form every cell type of the fetus. Therefore, they are pluripotent. Okay. Now, these trophoectoderm cells which were here, they can just give you chorion. So this means that they have already differentiated. They are out of that situation where they can become something else. So they have lost their pluripotency. Making sense or no? So what we are saying that this is the early divisions. They are actually the first differentiation division, if you think like that. Because in this division, already the differentiation has taken place. The outer trophoblast cells, which are trophoectoderm cells, are going to give you the chorion. They are not going to contribute to any part of the developing fetus. Only the inner cell mass is going to contribute to development of the fetus. Every cell will come, but they cannot give the trophoectoderm. But at the 8 cell stage, when the cell was like this, at the 8 cell, every cell had the capability to give you either trophoectoderm or inner cell mass. So if you have this cell, you can make a new human being. What do you mean by making a new human being? Because human being can only be formed if the placenta is formed. Making sense? So if that is not formed, if you take this cell, you transplant, say it can grow whatever you want to grow, but it's not going to give you this. If not this, then you will not have a new organism formed. So that's why these cells, up to 8 cells, are called totiopotent. Whereas the cell in the ICM, they are called pluripotent. They are not totiopotent. They can give you every cell type, but they cannot give you the whole organism. Making sense so far? So talk about this for a minute, then we'll go further.
Oxygen and nourishment from the mother. Secreting hormones to fertilization towards the embryo, modifying the mother's immune system, centering neural tissue rejection of the embryo. So the 8 to 16th division is the 4th division overall, but it's the first differentiation division. And generated cells, the generated cells are going to be grown, so no more part of the developing embryo or the formation of the cells that are going to develop into the fetus, which is the embryo cells. And these cells can give every form of any cell except the trophoectoderm, making them pluripotent. And then the other cells only get the trophoectoderm.
Wait, so Heather asked who wants to be out over here?
And then if the placenta doesn't form, it's not part of the individual.
Any doubt? Any doubt here? You guys are okay? So now, why to become trophoblast or trophoectoderm or ICM. So the decision to become the trophoblast or ICM. So the decision to become trophoblast or ICM is the first binary decision of mammalian life. This is interesting. You will say, even before you have become embryo, you take this decision right there. That which of those cells I'm going to leave out so that those cells can provide me nourishment. And the rest of the cell I'm going to utilize to make a new embryo, right? So then we say, okay, if this is a decision, then we have to think about what are those factors which are going to determine this decision, okay? So then we are saying the first differentiation take place, and you either get trophoectoderm or you get ICM. We are saying that ICM are pluripotent cells. Okay? So first we have to understand what is going to keep the pluripotency, and then we'll talk about how this decision is made. Okay? We'll talk about the things here. So then, if you want to have a pluripotent cell, the pluripotency is governed by three key transcription factors. So there are three key transcription factors which are responsible for pluripotency. Those are OCT4, SOX2, and the third one is NANOM. If you take these three transcription factors and you put those transcription factors in the skin, you can even change skin to the stem cells. That's what we call induced pluripotency. We'll talk when we'll come to the stem cells, okay? But these three transcription factors are the key to stay there as a pluripotent cell. So what these happen? These are transcription factors. They can bind to their own genes and the enhancer of each other to activate each other as well. So we know what are enhancers and all those things, right? Enhancers are the DNA sequences to which certain protein can bind. They facilitate the binding of the RNA polymerase II there so that their transcription can be activated easily, right? So those are the enhancer sequences. They make the loop enhancers and the suppressor and all those things in the DNA. And I think we did cover a lot about those hormone structures. If you're not familiar with enhancers and suppressors, so please go and read about those things a little bit, right? But these are the sequences on the DNA to which these proteins can bind. And ultimate goal is to bring the polymerase there, the RNA polymerase there, so that you can have the transcription. Okay? So then they bind. What they do, how they do it. So you have this SOX2. SOX2 can form a dimer with OCT4. SOX2 and OCT4 can form dimer. Now SOX2 itself as a transcription factor can bind to its own gene, activate more and more SOX2. SOX2 can bind to its own, OCT4 can go and bind to its own enhancer, activate more and more. Now, when they form a dimer, what they do, they can form dimer and they can actually activate nanome. They can activate nanome, okay? When the nanome is activated, what nanome can do, nanome is activating, is making them more and more nanome. Now this nanome, it can actually go back and activate itself. So now you have SOX2, you have OCT4, and you have nanome. When they are activated, what you get is you are ultimately maintaining, you are activating embryonic stem cell genes. You are activating embryonic stem cell genes. So, we are saying, we didn't still talk about how that decision occurred, right? We are saying that is the critical decision, that whether you want to become ICM or you want to be trophorectoblast. But we said to be ICM, to be inner cell mass, you should maintain the pluripotency. What is stem cell? Stem cells, this is important to understand. Stem cell means those cells which are not differentiated. That's the first thing. If they differentiate into something, then they are not stem cell. For example, this trophoectodon, they become something. So when you say maintaining pluripotency means you keep them in a stage where they are undifferentiated. And undifferentiated means that their chromatin is open. It is not going toward one side or other side. That means they are totally hypomethylated, but they have the capability to transcribe every gene which can give you every cell type of the body. So that's why we are calling. So that is what stemness. So that is called stemness. 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 So stemness is that characteristics that they remain undifferentiated. And when they remain undifferentiated, only then they can be pluripotent. We are saying these three genes, they keep them undifferentiated. So far it is okay. So now, what time is? Are we out of time?
Yeah.
Okay, so we'll just go over this and then we'll. So what we are saying that either you become ICM and trophoectoderm or trophoblast. That's the first decision. How that decision occurs we'll cover on Friday, right? But we are saying to have a stem cell characteristics, you need to maintain 3 transcription factor at very high level. Those are OCT4, SOX2, and nanome. And once they are in a cell, what they do, they can bind to their own enhancers and activate their transcription. They can also bind to enhancers of each other, and keep on activating each other gene. And what they do is, their purpose is to keep the cell away from differentiation. So these cells can divide indefinitely, they're immortal cell, but they are prevented to be differentiated. If you give a proper stimulus, for example, you're doing stem cells in the lab, once they differentiate, they become neuron. Then you cannot change them back to the stem cell. They are stem cell till they are undifferentiated. But you give a stimulus, like you're putting retinoic acid, you're making them to neuron or glia. Same way you give DMSO, too much DMSO, they will become cardiac cell. But before that, they are not differentiated. So that's what these 3 genes are doing. OCT4, SOX2, and nanome. That's what, when you put these genes in a skin cell, they actually de-differentiate the skin cell. So skin cell, they are not any more skin cell once you start expressing these genes. So they become undifferentiated cells. So then you can put them and make any cell. So that's what we will cover today, then we'll talk on Friday and go from there. Okay? Any question here? Okay, so when you